Second stage, mitosis. During mitosis, the cell nucleus divides into two new nuclei with set of DNA in each daughter cell. Now, again, we will repeat phases of mitosis: prophase, metaphase, anaphase, and telophase. What happened during prophase? Chromosomes condense. We mentioned before, the chromosomes thread-like structures, very thin, but during this stage, prophase, the chromosomes condense and one of chromosome attached to other to make one pair of chromosome. One or single chromosome called chromatid, which two chromatids attached to each other through centromere bind them together. Look at the one pair of chromosome, chromatid and chromatid, binded together by centromere to appear like duplicating chromosomes. Yes, duplicating chromosomes. The centrioles, second, the centrioles move to the opposite side of the nucleus. We mentioned in copying DNA and after that preparing for division. During preparing, the centrioles duplicate itself. One pair go far to the cell and the other pair go to the opposite side of the cell. So the centrioles move to opposite side of the nucleus. The nuclear envelope breaks down. The third nuclear envelope breaks down. So we have three steps in prophase. Chromosomes condense, centrioles move to opposite sides of the nucleus. Number 3, nuclear envelope breaks down. So what I mean by nuclear envelope breaks down means the nucleus disappears. No nucleus. Look at the shape. No nucleus. The two pairs of centrioles, one pair move to each side, and this pair of chromosomes lined up, attached to the centriole through spindle fibers, and no nucleus appear. Second stage, metaphase. Metaphase, chromosomes attaches to spindle fiber at its centromere and make one line. Number 3, anaphase. Look at anaphase stage. The centromere pull. The centromere pulls the one chromatid and the other one chromatid pulled to other side. At the end, the centromere cut the two chromosomes from each other. So anaphase, the centromere of each chromosome splits, pulling the chromatids apart. Each chromatid is now called a chromosome. This is called the chromatid and also called one single chromosome. This one chromosome or one pair of chromosome consists of two chromatids. After splitting, it appears like this. Each chromatid is now called a chromosome. At the end of the mitosis, telophase, fourth stage or fourth phase of mitosis, new nuclear envelope form. New nuclear envelope form like this to surround the set of DNA in each cell. So we have nucleus with a set of DNA. We have a nucleus with a set of DNA and one pair of centrioles and one pair of centrioles. Yes, and cytoplasm start divide itself to surround the nucleus in each cell. Yes, this is happened during cytokinesis, the last and third stage. The final stage of cell cycle, the cytoplasm divides. The structures are then distributed into each of the new cells. Each structure, cell with nucleus with a set of DNA and organelles, distribute itself also between the two nucleus. In this one we have mitochondria, we have lysosome, we have ribosomes, we have ER, we have cytoplasm, and so on. Yes, and the Golgi apparatus also in each cell. In each cell surrounds the nucleus. And the cytoplasm surrounds this jelly liquid fluid surrounds the nucleus. So at cytokinesis, Cytokinesis, the third stage and final one. The structure are then distributed in each of the new cells. When cytokinesis is complete, each daughter cell has the same number of chromosomes as the parent cell. At the end of this stage, each cell interphase again and repeat the cycle again, because it's called cycle, so it repeats itself. So one cell becomes two cells, and two become 4, and 4 becomes 8, and 8 becomes 16, and 16 becomes 32, and 32 becomes 64, and so on, and so on. This is duplicating for or division for parent cell. Start by one cell and then the number increase by cell division. What is the difference between cytokinesis in animal cells and cytokinesis in plant cells? The difference we mentioned before, the structure of animal cell differs from the plant cell, because plant cell surrounded by cell wall. This is rigid. Cell wall cannot squeeze like cell membrane. Cell membrane doesn't, animal cell doesn't contain cell wall, but contains cell membrane, which squeezes easily and allow splitting easily between the cell, and cell division happen quickly. But at plant cell, the rigid cell wall cannot squeeze together, so a structure called cell plate between the two nucleus across the middle of the cell begins to form a new cell membrane. Between the cell begin to form a new cell membranes between the two daughter cells. So the difference between cytokinesis in animal cell and in plant cell plate forms across the middle of the cell, and this begins to form a new cell membrane between the two daughter cells. After that, new cell walls form. After forming cell plate to separate between two new cells, at the end cell wall start to build itself. Cell walls then form around the cell membrane. But in this stage in animal cells, the cytoplasm benches into two cells. Each daughter cell gets the organelles of the parent cell. This is the 3 stages of the cell division. Interphase, stage 1, second mitosis, third stage cytokinesis. The longest one is interphase, takes almost 21 hours, but cell division, the 2 stages of cell division, the last 2 stages, mitosis and cytokinesis, both of them take 1 hour only.